Have natural history museums become irrelevant? Have their displays become so outdated and archaic that they are now misleading the public? Have museum curators consciously withheld certain fossils from their public displays? Fossils that would place the theory of evolution in jeopardy? Or do they not have the financial resources to update their museums? This man has raised these questions and asks for you to decide the answers. Life. It is wonderfully beautiful and amazingly complex. But where did life come from? How did we get here? Was the world created by God? Or is life simply a byproduct of a natural process? Evolution, the Grand Experiment, is a provocative new series that stares unflinchingly into this mystery and asks the most important scientific question of our time: what is the truth about evolution? Have the courage to question. Experience the thrill of discovery. Be a part of the Grand Experiment. This man has a true and unusual story to tell. It all began with a bet he made 32 years ago when he was just 19 years old. Since then, he has traveled the world to answer the challenge, to settle the bet, taking him to places he never dreamed he would go. Places like the Virgin Islands and the Great Barrier Reef. While diving at some of the more celebrated coral reefs of the world, he has witnessed an amazing array of underwater life, such as octopus, squid, sea turtles, and giant sea clams, large enough to swallow a small human, as well as a variety of other sea animals, such as feather stars, sea squirts, corals, and sponges. He has seen the majesty of nature on land. An Australian sea lion pup receiving a meal from its mother. Elephant seals playing in the surf in California. And wild bears feeding on salmon at a remote stream in Alaska. Reindeer caribou lounging in an Alaskan glade. Elk and bison reveling in a May snowstorm in Yellowstone. Koalas resting in the eucalyptus trees on Kangaroo Island. And kangaroo frolicking in Australia. This man is Dr. Carl Werner, an emergency room physician in St. Louis, Missouri. He makes his living repairing cuts, setting broken bones, reviewing MRIs, and at times consoling families. He is a dedicated physician who takes his job seriously and enjoys helping others. But when his day's work in the emergency room is finished and his free time begins, he turns his attention to his passion, finding the answers to his questions. Where did we come from? Were we created? Or did we evolve from bacteria billions of years ago, as Charles Darwin suggested? Is the theory of evolution scientifically valid? Or is it simply a myth? When Dr. Werner was a very young medical student and subscribed to Darwin's ideas, a fellow classmate challenged his beliefs and bet him that he could not prove the theory of evolution. And so his journey began.
A med school buddy of mine said to me, "Carl, here are several reasons why evolution can't be true." His arguments seemed plausible, and that really bothered me because I knew evolution was true.
Dr. Werner's classmate suggested that certain biochemical formulas and laws of physics contradicted the theory of evolution and the Big Bang. He asked how the universe could form naturally out of nothingness, since matter and energy do not form from nothingness. How could life begin naturally from chemicals if DNA and proteins do not form naturally from chemicals? And with hundreds of millions of fossils collected so far, why are so many crucial fossils still missing? The very ones that would demonstrate the transition from one animal type into another.
My buddy's challenge kept eating at me. I just couldn't put it down. So I decided to review the theory of evolution from top to bottom.
Dr. Werner was no stranger to science and hard work. In high school, he carried out an elaborate 2-year research project at the University of Missouri, examining the effects of a food preservative. This project and the following 6 years of study, leading to graduation from medical school at age 23, would prove to be just a warm-up for what would be his next major undertaking: evaluating Charles Darwin's theory of evolution. Dr. Werner studied the work of prominent evolution scientists, starting with Charles Darwin and moving forward through time. He read hundreds of evolution books, both historical and modern, plus relevant scientific articles. Finally, after 2 decades of preparation, he designed a practical experiment. To test the theory, Dr. Werner would examine the fossil record and look for evolutionary change. In order to keep his experiment manageable, Dr. Werner chose to focus on the fossils found in the rock layers associated with dinosaurs, the Triassic, Jurassic, and Cretaceous layers. The theory of evolution leads us to believe that animals and plants have changed over time. Dr. Werner's hypothesis predicted the opposite, that if evolution is false and organisms did not change over time, he should be able to find modern animals and plants such as ducks, dogs, apple trees, and roses in the older rock layers. This was a bold prediction, since Dr. Werner was unaware of any modern animal or plant species having been discovered in any dinosaur rock layers. Undaunted by the prevailing assumption that modern animals did not live with dinosaurs, he and his photographer wife, Debbie, set out on a most unusual experiment. Traveling to museums and dinosaur dig sites around the world, studying and filming fossils to see if life has clearly changed. Little did they realize that they would eventually travel more than 100,000 miles to 3 continents.
Wasserform, haben ja eine kurze Zeit gelebt, aber muss ein bisschen Sauerstoff da gewesen sein, dann ist es wieder eingegangen. Es konnte sich also
Here at the Solnhofen quarry near Frankfurt, Germany,
generell war es ein Lebensfeind.
quarry workers prepare stone tiles that are shipped all over the world for building materials. But this is more than just a tile quarry. It is arguably one of the most important fossil dig sites in the world. Over the last century, quarry workers and scientists have found tens of thousands of fossils here, including this dinosaur called Compsognathus. Many of the fossils found at Solnhofen are now housed at the Jura Museum in Eichstätt, just a few miles away. These fossils proved to be quite a treat for Dr. Werner.
I just couldn't believe the detail in the fossils at this museum. They were simply beautiful. The level of preservation allowed for a thorough side-by-side comparison with modern animals.
One particular fossil, however, caught his eye. A fossil shrimp. With its two very long antennae, it looked similar to the common Gulf shrimp in the United States. He wondered if this particular dinosaur-era shrimp could be a modern species and be evidence against evolution. Shrimp thrive in the brackish bayous of Louisiana, near the Gulf of Mexico. Local residents seine for shrimp as a way to make extra money, or simply to eat fresh seafood. After studying the photograph of the fossil shrimp and comparing it to a modern Gulf shrimp, Dr. Werner noted that both have long antennae, both have 10 legs, both have a segmented tail, and both have a prominent split bill above the eyes, thus sharing many common characteristics. Surprisingly, the fossil was assigned to a different genus, even though both shrimp share many common characteristics. And they were assigned to a different species, even though both look nearly identical. Dr. Werner found this odd. An animal's binomial scientific name is comprised of a genus name and then a species name. For example, a dog belongs to the genus Canis and the species Familiaris. Its scientific name is Canis familiaris. A species can be defined as a group of animals or plants that can breed and produce fertile offspring. Even within a single species, such as dogs, there are great variations in body size, shape, color, and types of fur. Besides these external differences, there are great variations in the internal anatomy of dogs, such as their skulls. For example, the skull of an Irish wolfhound and the skull of a bulldog are tremendously different. Yet these two animals can breed and are the same genus and species. The same holds true for the modern Gulf shrimp. Within a single species of Gulf shrimp, there are large differences in anatomy, including the positioning of the eyes, the shape of the bill, and color, but all can reproduce. Knowing of these variations within a single species, Dr. Werner questioned why the fossil and living shrimp were assigned completely different genus and species names.
To me, these two shrimp look more alike than two dogs, which we know are the same genus and species. I wondered if the scientists who named the fossil got it wrong. Could this fossil shrimp be evidence that animals did not change over time?
Dr. Werner continued the process of photographing and videotaping fossils from dinosaur dig sites and museums. then searching for the closest modern living relatives to see if animals and plants have significantly changed over time. In all, he and his wife took 60,000 photographs. Some of the filming locations were a tropical paradise.
Today I'm diving in St. Thomas in the U.S. Virgin Islands. I'm trying to find living organisms that look similar to the fossils that we found in museums that were taken from dinosaur rock layers.
Look at this pencil sea urchin tucked down between the rocks just a few feet from shore. It reminded me of a fossil I had photographed in Australia.
This fossil sea urchin on display at the Melbourne Museum was discovered in a dinosaur rock layer in England. It has two parts: short stubby pencil-like spines and a round body. The modern pencil sea urchin living in the Virgin Islands also has short stubby pencil-like spines and a round body. The fossil sea urchin's scientific name is Hemisideris intermedia. The living pencil sea urchin is Eusideris tribuloides. Again, Dr. Werner questioned why this fossil was assigned a completely different genus name, which suggested that the sea urchins have distinctively changed since the dinosaur period. Is it possible that modern sea urchins lived alongside dinosaurs? The Great Barrier Reef, located several miles off the coast of northeastern Australia, is the world's largest reef system, stretching over 1,000 miles, and is known for its corals and sponges. Dr. Werner dove here, looking for a match to a fossil coral he had seen at the Carnegie Museum. Before reaching his destination, this particular dive produced a treat. Dr. Werner was greeted by a giant 4-foot humphead Maori wrasse, who surprisingly performed a dance with this reef explorer. After this exhilarating encounter, his search continued for a modern coral to match this fossil exhibited at the Carnegie Museum. He eventually was successful. Although the fossil, with its round shape and mouth-like slit opening, was broken, the similarities between the living mushroom coral and the fossil were remarkable. But again, the dinosaur-era fossil was given a different genus and species name. Dr. Werner found fossilized examples of all of the major invertebrate groups or phyla living today in the dinosaur rock layers. He found examples of all of the major crustacean groups living today, such as crayfish, crabs, shrimp and lobsters. All of the major insect orders living today. All 5 major classes of shellfish living today. All 3 classes of sponges living today. Modern appearing corals. All of the major echinoderm classes living today, such as starfish, sea urchins and brittle stars, and both freshwater and marine segmented worms. The similarities between the fossil and living invertebrates were evident and difficult to ignore, yet each fossil had been assigned a different genus and species name.
I kept thinking about the scientific naming of animals and plants. Could this process be flawed? Typically, a fossil is assigned a genus and species name by the scientist who discovers it. Most of these scientists ascribe to evolution. Have they created the illusion of evolution that animals and plants changed over time simply by changing their names?
Dr. Werner began to believe that the invertebrates provided evidence that evolution did not occur. But would he be able to find examples of modern-appearing vertebrates, more complex animals with a backbone, at dinosaur dig sites as well? Or at this point, would his test of evolution fall apart? At Dinosaur Provincial Park, located in Alberta, Canada, 10,000 dinosaurs were buried over a 5-mile area, along with many non-dinosaur fossils. The Werners came to this spot looking for any modern-appearing vertebrates, reptiles, amphibians, birds or mammals that may also have been buried with the dinosaurs. After excavation, fossils from Dinosaur Provincial Park are brought to the field station for inspection. Some are placed on display for the public to see. It was here that Dr. Werner saw a fossil alligator called Albertochampsa langstoni. Initially, he compared the fossil to a living alligator, and they looked similar. Next, he compared the fossil skull to the bony skull of a modern alligator. Again, they looked alike, except for the fragile nasal bone, which was missing on the fossil. These similarities raised more questions for Dr. Werner.
Which pair of skulls looks more alike? These two alligator skulls? Or these two dog skulls? We know the dogs are the same genus and species. Why then was the fossil alligator given a different genus and species name?
Eventually, Dr. Werner found examples of all 4 reptile groups living today: crocodilians, snakes, turtles, and lizards in the dinosaur rock layers. In each case, when he compared the modern reptile to the fossil, they superficially looked as if they could be the same species. In some museums, such as the Milwaukee Public Museum, the curators did not display the actual fossils. Rather, they reconstructed what they thought the dinosaur-era animal might have looked like, based on the fossil bones. Since skin colors and patterns are not preserved in a fossil, the authenticity of the colors chosen by the museum's artists are debatable and unreliable.
I predicted that if evolution was not true, I would find the modern reptiles in the dinosaur rock layers. And astonishingly, I found them all. I found the crocodilians, I found the lizards, I found the snakes, and I found the turtles. Amazing.
Bernissart, Belgium. A small town near the French border was the burial ground for a herd of iguanodon dinosaurs. The Werners visited this location, widening their search for more modern-appearing vertebrates that may have lived alongside dinosaurs. In 1878, Bernissart coal miners found 29 iguanodons. Although the mine entrance is now capped, it is still clearly marked. The curator of the Iguanodon Museum describes the discovery of the dinosaurs in the late 19th century.
When the miners found the iguanodons, they had to transport a few blocks of clays containing the fossils here till 322 meters deep here in the pit. It was possible to reconstitute 29 fossils of iguanodons.
The second point that was very important, because at many places, okay, they found dinosaur bones, but nothing else. And here they found together many thousands of fossils together in the same layers as the iguanodons.
One of the fossils discovered here was a fossil salamander. Its appearance is uncannily similar to this modern Amazon salamander.
During the Mesozoic era, the age of dinosaurs, there were many animals that would be recognizable by people today. There would have been alligators, there would have been crocodiles, there would have been soft-shelled turtles, garfish swimming around at that time, lizards, frogs, salamanders, all very similar to the forms we see today.
This drawing of a Jurassic frog on display at the Redpath Museum in Montreal was based on fossils found in a dinosaur rock layer. Museums rarely display actual fossils of dinosaur-era frogs. Nonetheless, the similarities between the fossil frog and the modern frog are undeniable.
In fact, one of the pieces of evidence that comes out of this research is the recognition that amphibians, frogs and salamanders, went through the Cretaceous-Tertiary boundary in, again, our sample area in North America. There's no change.
Did modern types of fish coexist with dinosaurs, plesiosaurs, and mosasaurs? Paleontologists at the New Mexico Museum of Natural History and Science found this fossil shark tooth in a dinosaur rock layer. The similarities between this tooth and a modern goblin shark are undeniable. Dr. Werner questioned why this fossil was given a completely different genus name. Dr. Werner found many more examples of modern fish found with dinosaurs. This modern coelacanth looks a lot like this fossil at the Harvard Museum, but once again, the fossil was assigned to a completely different genus and species. Angel sharks live in the oceans today. This fossil angel shark was found with a dinosaur in Germany. Port Jackson sharks reside in the South Pacific. This fossil shark on display at the Carnegie Museum was found in a dinosaur layer. Guitarfish live off the coast of California. This fossil guitarfish was found with a dinosaur, Compsognathus. Herring live in the Atlantic and Pacific Oceans. This fossil herring was found in a Cretaceous layer in Lebanon. Along with this fossil eel. This bowfin on display at the Milwaukee Public Museum was found near a T-Rex at Hell Creek, Montana. When compared to the living bowfin, they look similar, except for the colors and skin pattern added by the artist. Flounder have been found in dinosaur rock layers, but these and other common fish are rarely exhibited in museums.
Sturgeon and paddlefish and alligator gar are forms that have been found in association with dinosaurs.
But certainly the stingrays went through unaffected, the chimerids, many of the bony fishes, things like the salmon, the salmoniforms, they've gone through unaffected, and today they give us a bounty of some of the best food we can eat.
Scientists have also found fossils of modern jawless fish, such as lamprey and hagfish. And according to evolution experts, they look almost unchanged from the modern forms. Again, these fossils are rarely, if ever, displayed.
All three groups of fish living today, that's the bony fish, the cartilaginous fish, and the jawless fish, have been found next to the dinosaurs, and they look the same.
Some of the bones of very big herbivores. Up here you'll see a section, you'll see leg bones, and here's some of the plant material I told you about.
Dr. Werner's hypothesis predicted that if evolution is false, he would find remains of modern birds at the dinosaur dig sites. But the thought of finding a duck or a flamingo or an owl at a dinosaur site seemed preposterous even to him. Still, he persisted with his experiment. Of the 10 dinosaur dig sites and 60 museums he visited, only one museum, the Milwaukee Public Museum, showed evidence of a modern-appearing bird displayed with a dinosaur. This bird, called an avocet, was found at the Hell Creek dig site in Montana. It is actually a reconstruction based on the fossil bones. If one disregards the colors of the feathers chosen by the model's artist, it is strikingly similar to the modern avocet. Armed with this sliver of evidence, Dr. Werner began interviewing scientists, asking if they had ever seen modern-appearing fossilized birds at dinosaur dig sites. He reasoned that maybe they were found, but simply not displayed for the public to see. The answers he received were startling.
What's becoming apparent is that many of the modern bird groups, parrots, maybe even penguins, and other kinds of groups like owls, evolved earlier in the dinosaur era and were beginning to pick up their traces. What we have so far are fragments of skeletons. We do not have completely articulated skeletons that we would say look like an owl or look like one of these modern bird groups. We have somebody that is judging on the basis of a limb bone that it really does look like an owl. I think this is an owl. And the evidence is circumstantial. It's building, but it's still circumstantial.
One of the exciting bits of information that's coming out now, one of the students here, Tom Stidham, has been going through our collections from the late Cretaceous, looking for the remains of birds. And what they're finding is that there is at that time, in the late Cretaceous, quite a diversity of modern groups of birds. Tom has just had a little article published in Nature in which he describes a late Cretaceous parrot. There are more pieces of information out there that are now being described in papers that are being written or in review, showing that other kinds of modern birds were present in the late Cretaceous.
Other scientists have described finding fossils of sandpipers, loons, ducks, flamingos, cormorants, and albatross in dinosaur rock layers. In 1998, Dr. Tom Stidham reported in the prestigious journal Nature that all of the major modern bird groups may have lived with the dinosaurs, based on fossil and genetic evidence.
To find evidence that all of the modern bird groups living today were alive during the time of the dinosaurs was amazing.
There are 3 types of mammals living today. The first being the marsupials, which raise their young in a pouch and include kangaroo, opossums, Tasmanian devils, and koalas. The second, called monotremes, do not raise their young in a pouch, but lay eggs. There are only 2 egg-laying mammals living today: the spiny anteaters, also called echidnas, and the duck-billed platypus. The last and largest category of mammals bear live young after a prolonged gestation. These are the placental mammals and include humans and primates, hoofed mammals, bats, hedgehogs, carnivorous mammals, and rodents. Paleontologists and museums teach that when dinosaurs lived, mammals were rare, small, and unusual. They refer to this era as the Age of Reptiles. Dr. Werner predicted that if evolution is not true, modern types of mammals would have lived with dinosaurs. In order to get answers, he met with Dr. Je Shilo, curator of vertebrate paleontology at the Carnegie Museum. In this interview, Dr. Lowe revealed that mammals were, in fact, common in the dinosaur era.
Give me an idea how many mammal fossils have been found in the dinosaur layers.
There are about 280 different groups or genera of fossil mammals, and of this, the vast majority are just fragments.
How many complete skeletons from the dinosaur era have been found?
So altogether, we're talking about less than 100, but they only belong to about 12 or 15 or so different species.
The name, the age of the reptiles, is that a misnomer then, that it wasn't just reptiles?
In a sense, the age of dinosaur or age of reptile for Mesozoic is a misnomer. The Mesozoic is about a great vertebrate diversity. In addition to dinosaurs, you have mammals, you have lizards and their relatives, you have crocodiles and their relatives, and plus many other groups that have gone extinct never left any modern surviving relatives today.
Dr. Werner predicted that modern types of mammals, such as hogs and hedgehogs, possums, porcupines and platypus, would be found with dinosaurs. But despite visiting numerous dig sites and museums, he did not come across a single modern mammal species found in the dinosaur layer. He wondered if mammals, like modern birds, had been found with dinosaurs, but simply were not displayed. He turned to university scientists and the scientific literature.
You know, for years I've been told that the mammals during the time of the dinosaurs were small, about the size of a shrew, and they were strange looking. But the more I interviewed, the more I realized this was not true.
Dr. Tom Rich is the curator of vertebrate paleontology at the Museum Victoria in Melbourne, Australia. In 1997, he discovered the jaw of an unusual mammal named A. nyctos in a dinosaur rock layer. The fossil caught Dr. Rich off guard for two reasons. First, it was a placental mammal found in Australia, the home of the marsupials. And second, it looked similar to a modern hedgehog jaw.
What did the animal A. nyctos look like if we saw him, fleshed out and walking around? What did this mammal look like?
Well, as far as we can tell, based on the size of the jaws and a little bit on the teeth, if you're going to take a modern animal, you'd want to pick a hedgehog in southern China, which lacks spines on it. And that's probably the closest match you can make.
So, to clarify this, it wasn't a modern hedgehog.
I have published speculation about that, not very seriously, but I just pointed out how similar it was to hedgehogs, but I doubt it.
In addition to hedgehog-like mammals, other mammals have been found in dinosaur layers. In 1929, Dr. Gaylord Simpson from Columbia University found an opossum-like animal named Alphodon in a dinosaur rock layer which looks eerily similar to a modern opossum. In 1974, Dr. William Clemens found the teeth of a primate at the Hell Creek Formation in Montana. In 1984, scientists in Australia found the jaw of a duck-billed platypus in a dinosaur rock layer. Recently, scientists found a squirrel-like tooth at Dinosaur Provincial Park in Alberta, Canada, and shrew-like teeth. Other scientists found what they called a beaver-like mammal in the Jurassic dinosaur rock layers in Mongolia, which was about 18 inches in length. Paleontologists in China found a fossil mammal with a baby dinosaur in its stomach. The mammal, named Rapinomanus, would have weighed 30 pounds and looked similar to a Tasmanian devil. Paleobotanists have called the age of the dinosaurs the age of cycads, a time dominated by unusual non-flowering plants and non-flowering trees. Television documentaries have reinforced this idea. Dr. Carl Sagan told his audience of 500 million viewers that dinosaurs perished around the time of the first flower, implying that dinosaurs would never have seen a modern flowering plant or tree. Dr. Werner predicted the opposite. If the theory of evolution is not true, he should find modern flowering plants such as roses and rhododendrons and modern flowering trees such as oak and tulip alongside dinosaurs. In other words, plants should not have significantly changed over time. In order to get the facts about fossil plants, Dr. Werner went to the prestigious Royal Botanic Gardens in London. What he learned was totally unexpected.
Yeah, basically my whole approach to my science has been to take these fossils and to compare them with their living relatives. What I'm interested in doing is understanding the plants of the past and comparing them with the plants today. And the way we do that is really very simple. We just get as much information as we can out of the fossil record on the plants of the past, and then we make point-for-point comparisons with their living relatives. So we look at the leaves and we compare the leaves in detail with the leaves of the modern plants. We look at the fruits, we compare the fruits in detail with the fruits of the modern plant. We look at the flowers, we look at the pollen grains and so on and so forth, until we get a really good understanding of just how similar are these plants. And when we understand those similarities, we can say something about their likely evolutionary relationships. So that's the approach that I've adopted, and what that's done for us is it's given us an understanding of what were the plants like during the last phase of the dinosaur era. And what emerges is that they were pretty similar to many groups that we have living today. Dogwoods, tulip trees, magnolia, and so on. Walnuts, oak relatives, chestnut relatives, and so on. All of these groups that we would think of as being sort of fairly modern groups of plants, also being around really quite a long time ago.
Along with the flowering plants, Dr. Werner also found fossilized examples from all of the other major plant divisions living today, including cone trees such as sequoia and redwood, ginkgos, cycads, ferns, horsetails, and moss.
Which is pretty interesting. It just kind of looks like a knob, but I've seen other fossil...
In testing evolution, Dr. Werner predicted if evolution is false, he should be able to find modern animals and plants next to dinosaurs. To his surprise, he found fossilized examples from all the major invertebrate phyla living today, such as arthropods, corals, sponges, echinoderms, worms, and shellfish. He also assembled fossilized examples from all of the vertebrate groups living today, such as fish, amphibians, reptiles, birds, and mammals. Over time, he became aware of fossilized examples from all 7 major plant divisions living today, including flowering plants. Dr. Werner contends that each fossil and its modern counterpart look nearly the same, offering strong evidence that animal and plant life did not change significantly, with the obvious exception of some animal extinction. Evolution scientists would disagree and counter that all of Dr. Werner's fossil to modern animal comparisons do in fact show evidence of change and evolution. Dr. Werner is not willing to agree with that assessment. After photographing thousands of fossils at 60 natural history museums, Dr. Werner has 3 criticisms of these science institutions. First, most museums have fossils displayed in one section of the building while modern plants and animals are displayed in another. Because of the inability to see the two together, this arrangement could falsely lead the public to assume that animals and plants have changed over time. He believes museums should place the closest modern organism next to each fossil. The visitors could then draw their own conclusions about evolution and change. Second, Dr. Werner has also noticed that museums in general falsely project the idea of evolution by leaving out modern varieties of common animals and plants from their dinosaur dioramas. If shrimp have been found in dinosaur rock layers, why are they not placed in the dinosaur-era ocean dioramas? Lobster, salmon, guitarfish, flounder, angel sharks, and so on. The same applies to the dinosaur forest dioramas. If oak trees have been found with dinosaurs, why are they not placed in the recreations of dinosaur life? Where are the flowering dogwood trees, the ash trees? Where are the walnut, chestnut, and sassafras trees that Dr. Crane spoke of? As for the museum images of pond life, where are the soft-shelled turtles, the frogs, salamanders, crayfish, crickets, mayflies, katydids, and lizards? And what about the lakes? Where are the ducks, the loons, the parrots, owls, and flamingos, boa constrictors, box turtles, possums, hedgehogs, primates, the beaver-like mammals, squirrel-like mammals, duck-billed platypus mammals, and Tasmanian devil-like mammals? By leaving common animals out of these museum displays, it makes it appear that animal life has changed more than it really has, and creates false evidence for evolution. Dr. Werner's third criticism is that during the time of his experiment, the most important fossils, the nearly 100 complete fossil mammal skeletons discovered in dinosaur layers, were not available for the public to see at any of the 60 museums he visited. Currently, only 16% of the US population believes in naturalistic, mechanistic evolution. Dr. Werner came to his own conclusions regarding the theory. He cannot help but speculate whether even fewer people would believe in the theory of evolution if the misleading arrangement of fossils and animal displays in museums was made known. If the ambiguous scientific naming of organisms was revealed, and if the public was made aware that more than 430 species of mammals lived alongside dinosaurs. Has the theory of evolution reached its useful end and been overturned by Dr. Werner's simple experiment? Or do his findings simply suggest there is cause for reasonable doubt and that more work needs to be done? Do you agree with evolution scientists that the fossils shown in this program provide evidence that animals changed over time, as Darwin originally suggested? What do you think?
What you have just seen is but a small portion of our story. If you would like more information, visit the website thegrandexperiment.com, no spaces, or read the accompanying book, Living Fossils.